Hi. Hi, everyone. Can everyone hear me clearly? Yes? Okay. So, welcome everyone to Preface headquarters today. I'm Ellen. I'm one of the co-founders of Preface. Here we have our whole team here welcoming such a great event and all the familiar faces here today. So, we are very honored to be part of Nobel Heroes Hong Kong 2026 and to welcome school principals, educators, business leaders, parents, and also students from Preface HQ and also online over there to join us for this very special dialogue. At Preface, we empower learners to decode the future through AI, technology, and also innovation. So today we connect that mission with the biological code that shapes life itself. We celebrate curiosity, science, and human progress together here today. We are very privileged to welcome the true inspiration and Nobel laureates whose groundbreaking discovery has reshaped our understanding of the world. But before we welcome him onto the stage, please join me to welcome our student MCs of the day. And may I now invite our students from Preface, Ellie and Ethan, to come over to the stage to share about Professor team's achievements.
Hi everyone, my name is Ellie. I am a student here at Preface. I am also an aspiring chemist who would like to mix different chemicals together to see what happens. That's why I'm so excited today. Our guest is a real-life scientific detective. He won the Nobel Prize in 2001 because he figured out how cells divide. Hi, I'm Ethan. We learned that computers run on code, but our special guest here discovered the switch that runs life. He found a special protein that disappears and comes back to tell cells when to grow. Do you know how he found it? He didn't use a supercomputer. He used sea urchin eggs and clam eggs. He went to a lab by the ocean every summer to study them. Our guest is someone who finds an answer to his inquiry through simple things. He says the truth usually turns out to be simple. Does anyone in the audience know what he named his discovery? He called it cyclin. He named the protein based on the protein's cycles. It comes and goes. Professor Hutt's love for cycling also led him to choose cyclin. To represent the nature of the protein, his curiosity drove him to discovery. He teaches us that you don't always need complex equipment to achieve your goals. He believes that curiosity is an essential trait that a scientist shall embody. The curiosity to know why things are the way they are. Let's have a look at this video of Professor Tim Hunt explaining how his discovery led to his Nobel Prize.
I can win a Nobel Prize. Anybody can. And I think everybody ought to want to, too, because at least if you're a scientist, why wouldn't you want to find out the most important thing you possibly could find out? So the great Erwin Schrödinger of cat fame said, science is a game. And I absolutely agree with that. And it goes on, but a game with reality. You get to check. Fantastic.
Right. So innovation is really about curiosity, determination, and having fun with all the discovery. So ladies and gentlemen, please welcome the man who unlocked a lot of secrets in the nature, Professor Tim. Yeah.
Okay.
And the moderator today will be our CEO and founder from Preface, Tommie Lo.
Thank you very much.
Wow, great! What an opening from our students. Great.
Yeah, terrific. Well done.
Yeah. Just now,
I've never been called a scientific detective. I remember that.
but do you like it, how you are described as a detective?
Yeah. That sums it up quite well, I think. Yeah.
Great. Then it somehow slightly shape what I'm about to raise to ask the first question. Is going to be a dialogue, and for our guests, please feel free to prepare questions just in case later on.
Do interrupt if you don't know what I'm saying. You know, if I say something silly.
So I was prepared for my first question, what it takes to be a successful scientist, what it takes to be a successful detective of nature.
Well, I think I think we all agree that, you know, curiosity is the main thing. You just want to know how the world works. Actually, I could tell a little story, which I like very much. So the first time I ever went and taught in Woods Hole, which is a very beautiful place. You ever get the chance to go to Woods Hole, go. Because it's a sort of science summer camp where a lot of biologists and other people go through. Anyway, so my first year there, my lab, the next door lab, was occupied by old Stan Cohen. And Stan discovered epidermal growth factor, which is a very important growth factor. And at the time, he was kind of stuck. His wife had left him, his son was on drugs, and his scientific research wasn't going anywhere. And we played tennis. He was very good. We were very well matched because he'd had polio as a youth, and so couldn't run very well. So that tells you how good my tennis is. And anyway, I liked him very much, and he was great. And then he then went on to win the Nobel Prize for his work on the epidermal growth factor and its receptor. And years later, I hadn't seen him for years and years and years. We got on really, really well that summer. And maybe it was 10, 15, even 20 years later, I happened to be in Nashville, Tennessee, and he was at Vanderbilt University. I went to visit him. And to my surprise, he was 75 years old, still in a white coat, and I found him in the cold room. And I said to him, Stan, what are you doing here? You don't need this. Tim, he said, it's pain and suffering. Pain and suffering. I can't stand not knowing what the answer is. And I push key around, and every 10 years or so, I find out something that makes it all worthwhile. And I think that really absolutely sums it up, the pain and suffering of not knowing. Okay? Because you feel that you're within grasp of some important, you know, you have a problem in mind, and you think about it all the time. It's in the back of your mind. But it does take, in my experience, usually around 10 years to solve most of these problems.
So in cases around 10 years, so it's not only just about simple curiosity, it's about curiosity come with...
It's stumbling on something interesting and important, which you think that you have the ability to solve with the tools that are available at the time that you start out.
So just now you talk about the pain of not knowing the things you want to know. How long, in your experience, what's the longest pain?
Well, as I say, I mean, I think that maybe it's not quite as much as 10 years, but it's something like that, actually. You know, that, and you don't always know what it is you're after. I mean, my first real project, my very first project as a graduate student, and I should say, I asked my advisor, what should I work on? He said, go to the library and find a problem. Okay, so I did. And in those days, there were libraries, you know. And, I found a nice problem, and it didn't work for trivial reasons. I used the wrong kind of rat in a summary. So after 6 months, I had absolutely nothing. I mean, literally nothing had worked. The only thing was I'd sort of learned what was in each drawer in the lab and how to pipette and stuff like that, you know. So it was good. And then I went to a scientific meeting, and I heard 2 talks that really set the scene for the rest of my career. And one was included sea urchin eggs and red blood cells. And the second one sort of explained or tried to explain this effect in red blood cells, the coordination between heme and globin synthesis. So heme is the thing that is the red thing that makes your blood red, and it carries the oxygen, and it's carried by a protein. And what the first speaker had found was that if you took away iron, so you could no longer make heme because it contains iron, the synthesis of the protein stopped. And that was a very surprising result at the time. So it was coordination. You had these two separate components, and if one stopped, the other stopped, right? There was a feedback loop. And so I thought that was very interesting. And then the second talk was by a very famous scientist who discovered the nature of the mutation in sickle cell hemoglobin. And he said that the way it worked was that the ribosome started on the messenger RNA, went along the messenger RNA until they'd made enough of the protein to bind the heme. They'd synthesize the heme pocket. And then if the heme was there, it would go in and they'd carry on. But if the heme wasn't there, they'd wait for the heme and form a queue waiting for the heme to arrive. And that's they stopped mid. So this sounded very peculiar. And when I went back to the lab and we sort of discussed this, we realized that the evidence that the guy had presented, very famous scientist, was wrong. He just got it wrong. He misinterpreted his own results. So we decided to find out for ourselves how it really worked, right? And so, my PhD was done in collaboration with another student, and we found out it was all nonsense, actually. That wasn't how it worked at all. They never formed queues, although we did experiments to show that if they did form queues, our methods could detect them. And that was good fun. And and along the way, another thing that happened to me was one time we were in the habit of going to the pub for lunch. And I was in the habit of having maybe half a pint of beer or maybe a pint of beer, at lunchtime. So never got much done in the afternoons.
That was your graduate student years.
Yeah, and one time I let the centrifuge run too long. So I thought, being lazy, I'll analyze the results anyway. And I discovered as a result of that the hemoglobin has two chains called alpha and beta. They're almost identical, but not quite the same. The alpha chains, there were fewer ribosomes on the alpha chain messenger RNA than there were on the beta chain messenger RNA. And that was my first real discovery, completely accidental, all because I stayed too long in the pub. But when I saw the results, I was confident enough about our methods that I got the right answer, which I then repeated it and confirmed it. And indeed, then another funny thing happened. We misinterpreted what the cause of that was, as a result of which we were corrected in the literature. It's very embarrassing to make a mistake. I don't like to make mistakes in public, right? So a friend called Harvey Lodish, a professor at MIT, repeated the experiments, got the right answer. And so we became friends because, we had a common interest in how this thing worked, and he got it right and we got it wrong, but it didn't matter because we were both interested in the same thing. So that was good.
Yeah. So somehow during the discovery of being the detective, somehow you can stay at the pub, and it wasn't only painful, I guess.
No. There was lots of lots of other things at the same time.
So I would like to continue raising questions in three phases. First, we want to learn about your life as a learner. So as a learner...
Well, you're constantly learning, right? Always learning. You're never there.
So what's the latest thing that you're learning?
Oh, what? The
Anything related to AI?
I learned last week were two things. Quantum computing doesn't really work. It makes mistakes. It has to, error. And did you know it had to run to almost absolute zero? And it's terribly sensitive to vibrations. Just a car going past in the street can completely wreck the bloody thing. And they're impossible to program. They're very, very difficult to program. And they get the answer wrong. So maybe in 30 years' time it'll be great, but not right now. So that was the first thing. The other thing was interesting things about AI, which we'll perhaps come on to.
Yeah, and actually, Tim told me he is a big skeptic about AI, so that's why we brought it to the last. Okay, so, but in case we want to learn more about your life as a learner when you were young,
yep.
Who was your heroes?
My hero was Francis Crick, because he was very clever in a simple kind of way. The first thing I really learned from him was he went to lots of seminars. He didn't do experiments himself, but he chatted to people all the time, and he went to seminars. And very often, almost always, if he was in a seminar, he would ask a question of the speaker. And I was a little bit surprised because quite often I knew the answer to the questions that he asked. And it became very clear to me that the reason why this great scientist who discovered the structure of DNA asked questions was because he hadn't understood what the speaker was telling him. And he would ask the question to clarify his own ideas, to make sure he really properly understood what was going on. And I realized that, and I found myself later that if I was puzzled, so were 98% of the audience. And if I put on my hand and asked a question, that was good. And I've always liked it when, you're allowed to do that. And so in Cambridge, I set up a little group with a friend that we had seminars by the people who had the most interesting stories at the time. And it was a room rather like this, a bit smaller, but one whole side of the room was a whiteboard with pens that you could write on it. And the rule was you were not allowed to show slides. You had to do it all on the blackboard. And the rule also was that you could interrupt the speaker at any time. Absolutely. And these were fantastically popular talks because you really learned how people had discovered things and really understood what they were doing and saying because of that. And Francis used to come to these talks as well. And so occasionally I would have had dinner beforehand. It was an evening thing after dinner, sort of 8 o'clock in the evening or something like that. And, I would occasionally sit next to him at dinner and we would have chats about this and that. And I almost never had a conversation with him, but that he made some remark which clarified something he seemed to know better than I did what I was doing.
Wow.
You see what I mean?
Totally. So, just now you mentioned about your time in Cambridge. From some of your previous lectures, some of your previous interview, you mentioned your time in Cambridge actually really trained you to become a scientist.
Well, I think so, yeah.
How was it?
Well, like the first supervision of my life. First of all, the guy asked us, what did we all want to do? There were 3 of us and the faculty member, zoologist. And he said, what do you want to do? And so we all said, sir, please, we think we want to be researchers, which was what I guess my generation wanted to do. Now, don't ask me why we thought we wanted to be the researchers, because I don't know, but we did. That was, and we had a blissful, naive confidence that somehow it would be all right. We weren't.
Was it a popular choice back in 1960s?
In the early 60s, I think it was, yeah. And God knows why. I mean, maybe we watched. Well, we didn't have television in those days. At least I didn't. There was no internet, and I guess I sort of read the encyclopedia or something. And the stories of discovery. I remember at school, I was my first real hero actually was Madame Curie, because her story is, I don't know if you've read it, but it's very, very inspiring. I mean, I would recommend anybody who wants to be a scientist, the first thing you should read is about Madame Curie and her 2 Nobel Prizes. This is a bull. And then the next one was Alexander Fleming and the discovery of penicillin. So, I think one of the things you learn about, I'm a great believer in people's scientific biographies, because the interesting thing is not what they found out, but how they found it out, right? And these are often very interesting stories of, struggling to understand things and figuring out how to get at the truth.
Just now you mentioned about other heroes who find out their discoveries, but from different researchers, different successful scientists, do you find a common way of getting into the final discovery?
Yes and no is the answer. I mean, I knew in Cambridge, I knew a lot of Nobel laureates because they were thick on the ground, and some of them, Sidney Brenner was clearly extremely clever. But he could be very cruel, and he couldn't resist making jokes, and he didn't care if it was a joke at somebody else's expense. So he was really, really nasty to particularly women, and particularly young women, which I didn't like. Other people seemed a bit sort of slow and sleepy, like Fred Sanger, who won 2 Nobel Prize winners. If you ran into him in the corridor, you would think he was the janitor or maybe the gardener or something like that. He was very, very low key, but absolutely brilliant in the lab, And I once had the pleasure of hearing his technician, Bart Burrell, explain how they got to the final method for sequencing DNA. And it was just like a random walk in science, I mean, we knew about this enzyme because of our work on that. And then we knew about this other enzyme because we'd met it, 10 years before. And I wish I'd had a recording of that, actually. I mean, probably Fred, he wrote a brief memoir and annual reviews of biochemistry, I think. And I ought to go back and look at it to see what he says about that kind of thing. So I'm always interested in how people behaved and I think the only thing that I concluded was that almost all the laureates that I knew, really successful scientists, they all wanted to get at simplicity. They all had this idea that there was an underlying simplicity to nature, and if you unlocked the key, what looked very complicated actually turned out to have some simple thing. But getting at that simplicity was very, very difficult.
Searching for that simplicity probably take a lot of sophisticated hard work or knowledge in the background. So when you were since you mentioned that you were made a scientist, you were trained as a scientist back in the time when you were in Cambridge. So what environment, what kind of atmosphere by that time really nurtured you to become that?
Well, I mean, what we didn't realize at the time, students in Cambridge are very highly selected, right? We're supposed to be the cream of our generation, but we didn't feel that way. I mean, what I was convinced the first year that I was going to fail my exams because I came aware of how ignorant I was and how little I understood. And when they said, you've got a first-class degree, and in those days, firsts were quite difficult to get because only 5% of people got first. Now the percentage is much higher owing to grade inflation. I thought it was a cruel joke, but it wasn't. It turned out that being skeptical and aware, because I think one of the tricks is knowing what you know and knowing what you don't know. And it's those unknown unknowns, which are where the real gemstones lie in research, right?
Yeah. So, just now we kept on mentioning your time in Cambridge, in UK, but I know that you also spent some time doing your research in New York in the States, right?
Yeah.
And you mentioned that you kind of like how the lab actually run in the States.
Yeah, I like.
What was that?
It was more collaborative. In Cambridge we were supposed to be on our own. Well, actually we weren't, because of what I learned in America, and I came back and collaborated with another student, and we made much more progress because of that, I would say. That's how we solved our problem. But just chatting about things, constantly involved. I mean, the first summer I spent there as a graduate student, which was a sort of formative experience, which, by the way, came about because I once drank too much wine at lunch. So there's an emerging theme here, I think. So this guy, it turned out that I got on very well with the chair of the Department of Medicine of the Albert Einstein College of Medicine, which is in the Bronx in New York. And he said one time, what are you going to do when you finish your PhD? And I say, to hell with finishing my PhD. Couldn't I come and work with you this summer? And he said, sure, why not? And by the time I'd taken a slow train back through Yugoslavia to Cambridge, there was an airline ticket awaiting me for New York. So I spent 3 months in his lab my second year as a graduate student. Then it was wonderful. We didn't find anything out, but I did find that that's where I wanted to go as a postdoc, because we were still struggling with this heme problem. We hadn't figured out how it worked. Later on, he became our major competitor. And actually, one funny thing was, this was the early days of the Science Citation Index, where you look and see how people cite each other's papers. And we became a peak. We were one of the most cited, highly cited papers because we would publish quite often and each cite each other's papers. And that was enough of an artifact to generate this gigantic peak of things. And it was absolute rubbish. I mean, we weren't the most important scene in town, but it looked like we were at the time, and figuring that out was how heme actually controlled protein synthesis. And it came about through other things that I learned while I was a postdoc. So it was rather peculiar. There was me and a technician, and that was pretty much it. And Irving, halfway through my time there as a postdoc, actually went off to MIT to found a new medical school. So he wasn't there. And we got stuck, or rather I got stuck. And after Grace went home at 6 o'clock in the evening, I started starting little projects on the side, and these were much more successful than the real one, and they were actually the things that unlocked the final secret, because I made a couple of discoveries that were really instrumental in unlocking the key finally. It was very funny. I mean, one was that something called, today they would be called, what are they called? Not reactive oxygen species, these fruit are supposed to have lots of antioxidants. So what are the oxidants? Well, somebody had an oxidant and it inhibited protein synthesis, and it inhibited in exactly the same way as leaving out the heme. So that was peculiar. Two very different things caused the same pattern of inhibition. And then I discovered that tiny amounts of double-stranded RNA, which is what happens if you have flu virus, for example, you get double-stranded RNA in your cytoplasm, and that's a signal to a cell that it's infected. And what I discovered was one of the mechanisms whereby a cell that it rings an alarm bell and dies rather than supports the virus growth. And I discovered one of the mechanisms by which it worked. It inhibits protein synthesis. This is also important in dementia, probably, it turns. I mean, all kinds of interesting things stem from this. So I kept on making these random discoveries, which turned out,
when you were in the States.
In the States, yeah. And, they got, and what I also discovered there was that, so to do poliovirus, there were people on the third floor I was on the sixth floor in medicine, and they were on the third floor in cell biology. And, we collaborated and did experiments together. So Ellie and I, found this inhibitor of protein synthesis, which puzzled us at first. It took us about 6 weeks to discover it was double-stranded RNA. And meanwhile, Nixon was bombing Cambodia without telling anybody. And so we used to go at weekends to Washington to protest, and I once got tear gassed right outside the White House and had to retreat to the Hilton Hotel in order to avoid they were choking gases. How anybody can survive tear gas, I just don't know. It was awful. And we could see the tanks lined up in the courtyards of the buildings around, which were ready to come out and shoot us down. It was pretty eerie, actually.
That collaborative spirit in the States, when it comes to research, did it really happen in Asia? Did you? Because I know that you spent some time in Asia as well.
Well, I had some good collaborators in Japan because they said the same thing. And I got a couple of wonderful postdocs from one of these people, and a very nice collaborator from another one, and also access to coming to Japan, which is something I was very frightened about. Because don't underestimate the power of propaganda. And when I was growing up, the Germans and the Japanese were the enemy, and the Germans were sort of Donner and Blitzen, Schweinhund, and the Japanese were unbelievably cruel and horrible. So when I first went to Japan, I was extremely nervous because, I had this racial stereotype, and it came as a real shock and an education to me to find out that the Japanese were just like us, except, culturally different and rather puzzling. And I wouldn't say that you can never say that you really understand the Japanese, in my experience. They suddenly explode for no known reason. It's very strange. But on the other hand, it was very productive. And the last postdoc I ever got was one of the best I ever had. And we made right at the end of my career, a very nice discovery and worked out what was going on because he was terrific. And not only was he terrific, but also we made a great team. And one of the problems when you get sort of rich and famous is that, you have to be able to criticize each other. And I say to Satoru, I think you should do this. And he would make a face, but then do it. But he was equally capable of saying, Tim, I'm not going to do that. That's a stupid idea. And and we were both...
Your postdoc
we
student?
were both fine. Yeah, and we were both fine with that because we knew that we were both on the track of something which was sort of bigger than both of us, right? And you had to be that critical of each other's ideas because we were trying to get at what the hell was going on. Easy peasy.
Great. So I'm going to my last question because I would like to reserve time for open to questions. It's actually quite leading to something what we want to mention about AI because I know you're a big AI skeptics. But also me too. We teach AI for a living, but then I will say I'm also skeptics about AI at times. But then you mentioned to your students that don't totally believe in what you read.
That was an important lesson very early on, actually. Don't believe what you read.
And it's on YouTube, so I actually check
it out for yourself. Make sure that your feet are securely planted on the ground before you take the next step. And this is really, really good advice. So you want you want your findings to be really robust because you're building everything on what you know. And if that's on shaky foundations, you're sure the building will fall down when it gets to a certain height, right?
Yes. So don't totally believe in what you read. It's actually one of the core or original scientific genes or push actually inside you, and how do you correlate it to what AI, the large language models, are producing on a daily basis?
I think that's interesting because the trouble with the large language models, as I understand it, and I don't claim to understand it very well, is that they're fed let's say the scientific literature. I mean, they're trained on something, and the literature is not all right. And how is the AI model to know what's right and what's wrong? I mean, I remember once having sort of being amused by some structural people who were trying to predict a structure, and they believed a paper that was completely wrong. So, needless to say, it completely messed.
So then AI studied it.
This was in pre-AI days, right? So, garbage in, garbage out, right? And I don't know how so if you have, what AI is very good at, I understand from those who use it, and I have no direct experience myself, I should say, is that protein structure prediction it's really, really good at. And it mostly gets it right if there's a structure if this protein has a structure.
Because the pattern is more predictable.
Because it's based on a very accurate, curated set of structures, which it interprets then. I mean, it's very good at looking for patterns and finding things out from the patterns, right? And if the database is 100% accurate, then its output is pretty accurate too. But if it's trained on just the real world ramblings of a fevered mind, it will probably produce itself equally fantastical results, which they call hallucinations, but just means they get it wrong.
Wow, great. So now I would like to open the floor for questions, and I'm pretty sure there will be a lot of interesting questions to Professor Hunt.
Professor Hunt, thank you very much for sharing. I'm Yanni. I'm a neurologist, a researcher. Yes, I totally agree with you. So using AI for research is so much difficult for us nowadays. But nevertheless, it has really accelerated the progress and development in many of the research areas. But while many of you might thought that AI would make many of our lives easier, it actually makes the lives of researchers and scientists more challenging.
Really?
Because we're working in areas of unknown.
Right.
So it's really difficult for us to find out what are the errors that are involved in all those data or results generated or processed by AI, because the process is often not transparent and it's so complex that we don't fully understand the algorithms involved. So, but nevertheless, it's a very powerful tool that we have to embrace in this era to augment our knowledge and our development. So in your point of view, how should we work with AI to ensure the scientific integrity of our research findings?
Well, I guess it's like any tool. You have to sort of test it and see what it can tell you, what it's good for and what it's not, right? And I was interested to hear from some of the AI people I talked to in Singapore. It's supposed to be really good at writing code, but they said it's really bad for writing code because although the code it writes works beautifully, you can't understand why it works. And that's something that's written down. I mean, you can read the code, but you don't know why it works, and therefore you can't maintain it. So it's very dangerous to write your program using AI. It's good if you use little short snippets, but not for great big programs. That was the message I got. But I'm not a computer programmer, so although my daughter is. And she said that we were talking earlier about vibe coding, and she disapproves strongly of vibe coding. I don't even know what vibe coding is.
Great. Any further questions?
Any other comments? Yes.
George Hong Choi. You got the Nobel Prize. At some point during your life, do you suddenly realize I'm a very smart guy? And you have two young students here. And when was that? And then was that a few years from then, or is at that point in time you had a breakthrough? What was that moment? Suddenly you realize you can achieve something.
Oh, I'm glad you asked that. So it's a long shaggy dog story, which you probably don't have time for. But basically, I've been worrying about why clam eggs and sea urchin eggs made new proteins after they were fertilized. Because the whole thing about eggs is that you lay down a store of all the things you're going to need early on. And then I sort of began to dimly appreciate that if you inhibit the synthesis of new proteins, the cells don't divide. And people had known that for a long time, but they hadn't drawn the very simple conclusion that that meant that one or more of these newly made proteins must be essential to catalyze cell division, because there were other explanations of why that might be, really. And so I did an experiment which was designed for something completely different, to compare patterns of protein synthesis in different kinds of fertilization, basically. And when I developed the gel, I saw that the most abundantly newly synthesized proteins, which you could easily detect at early times by my simple methods, suddenly faded away just before the eggs divided for the first time. Now, I had that result, and then that evening, I ran into a man whose seminar I'd heard 3 years before, who worked on the enzyme that catalyzed cell division. And I told him my result, and he told me a result which wasn't yet published, which was electrifying, which said that you needed new protein synthesis to turn this protein that he was to turn it on. So I knew within 24 hours of seeing this protein go away that I had made the discovery of a lifetime. Very unusual that. Normally you sort of gradually inch your way towards the truth, but in this case, bang, it was there. I knew. And so, I mean, the first thing I had to do was to repeat the experiment and see if it was really true. I hadn't been dreaming, and do the experiment better and time division. And it turned out the protein went away before division was completed. So that was good. Also that, it went on. As long as there were dividing cells, this protein kept coming and going, and things like that, and various other things, like if you inhibit DNA synthesis, so the cells no longer can enter mitosis. This protein was perfectly stable. So it was only unstable in a very narrow window just before the chromosomes came apart at the metaphase to anaphase transition. So 10 days later, I wrote a letter home to my collaborator in Cambridge saying I think I'd made, a very important discovery, and I had. So when it got back to Cambridge, I had to write this up, and I wrote it up and submitted it to a leading journal who one of the referees said, this is wild speculation based on faulty logic. Not the kind of... But the other referee was a guy called Paul Nurse, who later became my boss and is my and he said he thought that this was pretty interesting. So I had to rewrite. So the editor said, the good news, we will publish your paper, but the bad news, in nothing like its present form. So I had to rewrite the paper, but there were no more sea urchin eggs, and I was in Cambridge. So I couldn't do any more experiments. I had to use the very tiny few shards of information I had, and fortunately that was good enough. But if I read that paper, I'm very embarrassed because my head was somewhere completely different. You can see that. But right at the end, I say, that the behavior of this protein means it's got to have something to do with MPF, the enzyme, the factor that catalyzes mitosis. But so there was a real eureka moment. But I don't think that means that, it was still a terrific struggle then to find out what the hell it was and how it worked. And there we were really stupid. So I would say I was really pretty stupid all the way along. But so were some very smart friends of mine. It's sort of interesting how, again, prejudice blinds you, because in the end we found out what it was, and it was a kind of enzyme that was very well known. But whereas my factor turned the enzyme on, previously known example turned it off. It was that, how stupid is that? If something can turn something on, of course it can turn it off. There's nothing in theory that says that. But with why did it take from 1982 to 1988 to figure this out? Then there were lots of clues. But I think, again, Francis Crick once gave a talk I heard, and he said, when you're at the frontiers, it's all so foggy, and there are so many clues, and some of them are true, and some of them are relevant, and some of them are not, and you can easily get misled, both by the clues and by your, as you say, your prejudices. And I think it's mostly overcoming those prejudices they're sort of unexamined at the time. That's the problem. They're just there. You're unaware of them. So this is very difficult. And that's why the pace of research is usually very, very slow, because you're sort of feeling your way through the fog, and sometimes the fog clears, and more often it doesn't. So you're usually working in the dark.
So basically, by the time that you really make that discovery, you think that you're also quite a smart person.
No, on the contrary. No, I don't think, and at school I was never a very smart person. I never came top of the class or anything like that. So I have a pretty low opinion of my... The only thing I'm good at is spotting things that don't quite make sense. Okay, little clues that give the game away.
So it's a good full circle of going back to how to be a good detective.
Yes, exactly. Which clues to focus on. I guess. I'm not a great reader of detective fiction, but
Can I have one or two more questions?
Some two side by side back there.
Thank you so much for speaking to us, Professor Hunt. I have a question regarding your work in cyclins.
Yeah.
I'm sure now that they've all been sequenced to heaven and back, and you have a list of all the mutations. This links into the question of personalized genetic medicine. Do you think that with everything that's been sequenced already, we'll see any personalized medicine or DNA-based therapies that will be affordable in our lifetimes? There's so much at work already.
That's a good question. I understand you can now cure sickle cell anemia, for example, by putting in the proper DNA for an affected beta chain. But it costs $1 million. And I don't at the moment see that changing any time soon because it's really, really tricky. You should really be asking my wife, who is involved in gene therapy, this question, because I'm always teasing her that it doesn't really work, and she's saying, yeah, no, it really does, and it is true, that it was a kind of engineering problem. I mean, in theory, it ought to work, but there was an awful lot of work that had to be done before you actually got the practical details right, and they're beginning to get it right. Some diseases are easier to cure than others, and sickle cell anemia is pretty impressive, actually, if you ask me. Which is, by the way, what my lab in New York was trying to do. I mean, that was the dream, that you could do that. I remember my boss actually injected chicken DNA into a rabbit to see if he could detect chicken hemoglobin. This is long before cloning. I mean, it was a ludicrous idea. Anyway, but it was the right idea, but poor execution, I would say. So I don't know. I'd be sort of surprised if you could do it. It just takes a lot of people, a lot of time, and a lot of care to get right. But you never know. I mean, it's like DNA sequencing. It used to cost, what was it, £3 billion for the human genome. Now it costs you $200. Pretty impressive, and because the technology has changed completely from how it was when I used to do it. Here we go.
Hi, Sir Tim. It's really our honor to be sitting in the same room with you.
Oh, no, don't be ridiculous.
I was a cardiologist, but what I want to ask you is, I'm reading that a lot of immunotherapy, cancer immunotherapy, actually being moved ahead inside China.
Oh, really?
And even in other stem cells and all these. But I want to ask you, since you are a learned scientist, what do you think progress that mainland Chinese scientists are having, and where do you think they are now? For example, we are hearing that they are really catching up in AI with the West, but also in medical science. So I think they are getting somewhere. But I want to learn from you what your thinking is, and where do you think they can together contribute?
Well,
in this science world.
This is a very, very interesting question because So one of the people that I knew in Cambridge was Joseph Needham, who was very impressed by medieval Chinese science and technology and wrote a huge multi-volume work about it because he'd been a scientific attaché in Beijing during the war, I think. And, but the funny thing is that then stopped. I mean, it just became a body of accepted fact, and it never, for some reason, made any progress. And so it seems to have remained for a very long time. And I remember when I first went to Shanghai, to a university there, being really rather disappointed by the researchers I talked to. They weren't bad, but they were very much followers rather than leaders. That was probably 20 years ago or something like that, I suppose. I can't remember whether it was pre or post Nobel. But, anyway, now things have changed radically because it's not because Chinese are stupid or incompetent, but all the brightest minds had gone to America, and then they all came back to China. So now there are some fabulous labs in China publishing fantastic papers at the forefront. And I haven't been back to sort of talk to them and find out what's going on. But I know one or two of them. There was one guy in particular who we collaborated, who was very kind to us and helpful right at the end of my career. And, absolutely outstanding scientist. And he's now back in, I forget which, probably in Beijing, I think. But, so I think the future is very bright. And it's perfectly clear to me that, it's very peculiar how held back Chinese scientists were for so long. And I don't really understand why that is. There's something in the cultural water which is important. You need sort of role models. You need to free people. I mean, one of the problems I see more in Singapore, I think, than here in China is that governments demand an instant return on their research investment. And that's an engineering problem. You already know how it works. You just got to fiddle and improve. But the really important things come from probing the unknown. And certainly in Singapore, I was on a grants committee there, and it used to irritate me intensely because they were always looking for people. So the people in the labs would set up startups on utterly half-baked ideas. You could tell they were never, ever going to work. And that then distracted them from really asking nature how she worked. And that's what you should be doing, in my view. So one has to be very careful to distinguish these two kinds of research. There's a kind of research that gets gene editing and gene therapy going, which is, you know the principles, you just got to tweak it so that it actually works. And when you really don't know what's going on, which is most of the time, and you've got to ask nature directly how she does things, right? And let's say you don't know what you don't know. You know, there could be wonderful secrets just lurking behind in plain view, but invisible to our stupid preconceived notions of how things are. I think, I mean, you know, one time going to Lindau and hearing these physicists talk, and they were all very arrogant lot. I mean, they said, when we turn our laser-like attention on biology, you guys are going to be toast, you know. But it's not like that. I mean, you discover that actually there's something called dark energy and dark matter. Most of the matter of the universe, they don't have a clue what it is. You know, so this is amazing, actually.
Great. And one more question.
Yes, you've been wanting to for a long time.
Thank you for taking one more question. I'm a high school science teacher, history and biology specifically. And we've talked a lot about your university experiences and your research.
Yep.
But I'm always looking for ways to inspire my students to go on in science.
Yes.
So I'm wondering so before you started in the great university education career that you have, what inspired you as a young person?
Well, as usual, 2 school teachers.
Yes.
And the first one was when I was a little boy at prep school. We had endless Latin and Greek lessons and only one science lesson per week. But it was that lesson that I really looked forward to because Gert was a marvelous teacher, and he had insight into the mind of the 12-year-old boy. And he would make explosions and do simple demonstrations and ran a science club and stuff. And so we watched there in awe as he made radio-controlled gunboats, for example, that fired real torpedoes. And I don't know whether they exploded, but anyway, you know, I couldn't do that. It was electronics, and I had a friend who was much better physicist than I was. That was a very useful lesson. He taught me that I was good at biology and bad at physics. That was a very, very useful lesson. And then in secondary school, there were 3 of us in the class, and we all became biochemists in the end. And we would often like my pet rabbit, my brother's pet rabbit died. And so I took it into school and we dissected it, right? And that was a revelation because we'd only dissected the 7th cranial nerve of the dogfish before. And these were formalin-fixed dogfish. It was awful. Everything was sort of dull, browny grey color. And to see the bright colors of a freshly dissected rabbit was amazing, you know, and extraordinary. And then we sort of walked out onto the fields and said, you know, why is this plant growing here and not there? So I've always liked doing things rather than learning things. I'm not very good at learning things, but I love practicals. I love actually seeing for myself, I think. And so that's what I think that, you know, that was really, really helpful. And even in Cambridge, I did much better in the practical exams than in the theory exams. So I don't know what that says, but that's how it was. Yeah. But those teachers, those two teachers were really, really, really important.
Great. I think we had a pretty good dialogue, pretty good sharing from Tim, from Professor Hunt.
I'm sorry to be so incoherent. I get it.
No, sorry. I think it's we all enjoyed the session. We don't know that it's actually a session a little over an hour, but we didn't feel it because we very much enjoyed it. Thank you so much, and I'm Poppy. So feel free to take a few more questions just in case. Well,
Sure. I mean, I don't have to rush home.
okay. You know what? You're very dangerous of saying that. Thank you.